Okay, full house. Welcome everyone. Hello, and it's my greatest pleasure, Ian, to introduce Florian Engert. Hello, Ian, from Harvard University. It's a great honor to have him here again. I think last time you were here 4 years ago giving the talk. It was still in the midst of the Corona pandemic, and so Florian Engert studied in Munich at the beautiful LMU. Physics, and then decided to kind of drop physical systems and going into neuroscience to do his PhD with Harald Lux. Hans Lux. Hans-Dieter Lux on the leech serotonergic uptake, and then transitioned during his PhD to Tobias Bonhoeffer, where he was pioneering, one of the first people who actually used two-photon microscopy in a living sample, and develop methods to image in vivo from growing and shrinking spines, and was kind of showing for the first time that synaptic plasticity happens in the nervous system. Then dropped the rodent system, switched to Mu-Ming Pu in UC San Diego, to work on tadpole visual system, and developed kind of imaging methods also there to study synaptic plasticity. Then got faculty position in early 2000s at Harvard University, where he then also dropped the tadpole, but switched to a very similar system called the zebrafish, where he then continued that work and then pioneered methods for whole brain imaging and electron microscopy and fancy connectomics and all kinds of tools. However, is now realizing that all these technologies and these techniques, which neuroscientists are so obsessed with, belong into the supplement of all the papers because they are relatively irrelevant.
So he dropped all of that too?
Dropping all of this too, and now getting obsessively focused on behavior in animals with larval zebrafish. And now recently, so last night on the Christmas market, we talked probably for 5 hours about honeybees. So now he's probably also going to drop the zebrafish and switch into the honeybee. And on the side, he is, I think, a great mentor, which I experienced myself. I learned a lot, also to generate a positive atmosphere across scientists. And he's also having an excellent, I think, work-life balance. And he's going to show some photos of that in a second. And he's here until Wednesday, so there's still opportunities to meet. Tonight we go for dinner, and otherwise the stage is yours.
Thank you.
Thanks again for coming and looking forward to your talk.
Thank you, Armin. Thank you so much for the invitation. I've dropped the whole brain imaging and the connectomics. I'll talk a little bit about that, but I just want to pick up what Armin just said. I've become re-obsessed with behavior. And I want to restate that again, that I think if you want to understand how the brain works, if you want to be a real neuroscientist, then the critical question is that you need to really ask yourself, what problem is the animal trying to solve using its brain? And so I would say that the work that's being done by you guys here, and also by Ian, is the hard problem. This is the real question in neuroscience. What problems are animals solving, and how are they solving this? Which algorithms are they using to solve it? The neural implementation, I think, is just the control experiment. So I would say that you people, you ask the good questions. How it's implemented by the brain, I think, is more like is the control. So behavior, I think, is really where the rubber hits the road. And also not just what the animal is doing, how it's behaving, but which algorithm, which computations are being processed by the brain. What are the computations that have to be going on in order to get the problem solved? And once you have realized that, what are the temporal dynamics, the computations, the attractor states, if you know what the animal has to solve and which aspects it needs to keep in memory and keep to compute on, then finding the neural implementation turns out to be infinitely easier if you already have an intuition about what the computation is that's happening. Since I have a lot to tell you, I think I'm going to skip. Well, maybe I'll show you briefly. When Armin was talking about work-life balance, I just came back from a sailing trip around the world, the Clipper race. Those of you who are interested, Clipper around the world race, 6 months, a fleet of 11 boats, dedicated racing yachts. Each boat has a crew of about 20. And this is the 8 stages around the world. I did 3 out of them. I went from South Africa to Australia, around Australia, and from Australia up to China. It was awesome. Work-life balance. And those of you who are interested, talk to me about this after the talk. Today I want to talk about not so much the computations that animals do, but internal states. What motivates animals and what governs animal behavior when animals transition between different internal motivational states. Which I think is to a large degree neglected in neuroscience, that animals don't solve all problems in parallel. Before an animal engages the world, it needs to pick a problem, which particular aspect of the world it's interested in. So this is about internal motivational states. Pick your problem, the problem that you're interested in, and how is that governed by the brain? How is it decided which problem I should engage in? I think this has also to do with emotional states, and I will talk about today what we've learned in a larval zebrafish about these switches between internal states. This is sort of the overarching structure of the talk, and I'll focus in the first half on a specific problem that zebrafish need to solve and how they can switch in and out of that task. And in the second half, I will discuss what the role of the body is, how internal body states can actually influence your decision making. So this is listen to your heart. Here is where we'll talk about how heart rate can actually influence what an animal engages in. So turns out that all animals listen to their hearts all the time, even if they're not aware of it. So that's the general structure. Yes, so we'll use the larval zebrafish for understanding interactions between brain and the entire body. Here we'll have the heart beating, which you can see. Here is we can monitor brain-wide neural activity. Again, this turns out to be mildly informative, but not all that informative. It's certainly not the core. Where we can look at motor behavior as well. We can do this all at the same time. And the task that we will study in a larval zebrafish is the one of position holding in a moving stream. That's the workhorse of fish behavior. So here we have an animation of a larval zebrafish. And as you can see, it's hanging out in a current. And what you'll see is it manages to hold position in a moving stream. Turns out that that's actually not an easy problem to do, especially if the environment is noisy, if the flow information is noisy. This is not that straightforward in a noisy, complicated environment. But this is what we studied for the last 20 years, really, how fish solve this one particular task. And here's another implementation of that. On the left, you'll have a bunch of fish swimming around randomly, just so you know what they look like and how they swim. On the right, we'll have a larval zebrafish here that is looking at coherent dot motion. Now the motion has stopped. The fish is swimming normally. And you'll see when the motion starts again. So coherent noisy dot motion, wait for it. Here now, dots start moving and the fish integrates this information, looks at it, it's noisy, it's hard to see, but eventually he'll start following this whole field dot motion as if the fish is trying to hold position in a moving stream here. This is a sensory evidence accumulation and decision-making task that we can study in a larval zebrafish. It's relatively straightforward to implement it in the lab. I'm not going to talk today how this is done by the brain. What I'll talk about today, how the fish will choose whether to engage in this task or not, and based on what. So this is only one of the things a larval zebrafish has to do in life. And today we'll discuss how an animal decides whether or not to engage in a task like that. All right? Now here's again. That's the task. See it again. Here dots are moving, fish is trying to follow along. Actually, we should spend maybe a minute here on that because you'll hear a lot about that task in the next 20 minutes. So if that's not quite clear, then we should clarify this right now. Again, the challenge for the fish is to find out which way are the dots moving. Are they moving left or right? Once the fish is sure, it needs to make a directed turn into the direction of the dots. They do this innately, or they don't need to learn that. This is a reflex behavior. And here we test just how well they do that. Everyone okay? What the fish is doing? Why they're doing that is probably to hold position in a moving stream. You all good? I mean, that's not that exciting and interesting, I would say, as a problem in life, but we'll use it as a probe for, I think, more interesting and fascinating problems, and that is about attentional switching. So now we'll ask how good are fish at doing that task, huh? And ask if they don't engage, why not, and what's their problem. So again, here is one way to, just as a primer, what you've seen in the previous movie looks like that. And because in this case, we want to do large numbers of fish, we want to collect a lot of data. Turns out this is going to be really critical, that we can get up to 2,000 trials, like 3-minute trials per day. We need large data to extract the relevant statistics. So we had to scale up our experimental rigs. So here we see 1, 2, 4, 8 of these units. So we can look at 8 fish at the same time. And now we have scaled this up even more. 8 times 8 is a lot. I'm not going to do the math here. Yeah? And the task is relatively simple. The fish for 150 seconds looks at noisy, non-moving, just flickering stimuli. And then for 30 seconds, we show them a moving stimulus and ask every second he makes a decision. We can get roughly 30 choices and decision makings. And then we repeat this. So this is 3 minutes. We repeat this for 90 minutes. So we get 30 trials for a given fish. And because we have 64 fish in parallel, we can get about almost 2,000 trials in a day in 1.5 hours. So we can just collect a lot of data of that. It's just the background statistics, how we can get so many data, so many decisions. Huh? So each time that we are asking the fish to make a decision, there's noisy stimulus, and so we can do just a lot of trials. All right. That's the data we get. On the x-axis is the turning angle. On the y-axis is just a histogram. And what you see, if the dots are moving with 100% coherence to the left, then the fish is making a lot of correct decisions, yeah, into the left direction, -50. Yeah. Often they swim forward, meaning they don't do anything. They just don't give a shit, right? They just swim forward, and they make very few turns in the wrong direction. Okay? If we reduce the coherence, we make it harder for the fish, then you see that this psychophysics curve, then they make more mistakes. That's allowed. I would say we shouldn't judge the fish for that. We just make it harder, yeah? And then they have a harder time making the correct decision, so that's okay. Yeah? And then if we give them zero coherence, then we see a symmetric turn distribution, which also makes sense. Yeah? They're not psychic. And this is for the left and for the right. This all makes sense, yeah? This is our performance metric. Lots of 2,000 trials went into that. Now, where we are unhappy, where we do judge the fish, if we look at the performance score for left coherence and right coherence, if they make 100% correct decisions, they would be here up at 1. If they make only wrong decisions, they would be at -1. And what we find is that the fish don't go up to 100%. They go up to about 80%. So only 80% of the choices are correct. 20% are incorrect. And that we can judge at 100% because that's not hard. That's an easy task. The fish really should be able to do better than that. So we're kind of disappointed in our fish. So what's wrong with you? Why? And this is not just true for fish. You find this in almost all mouse, monkey, human behaviors, that even for an easy task, animals underperform. And I'll explain to you today why, if that happens to your children, to your spouse, to your partner, to your friend, to your dog, you shouldn't be all that disappointed. You shouldn't judge too harshly. There are reasons why animals don't perform perfectly, and maybe you'll get there already when you think about it. where I'm going with that is they underperform, not because they can't do any better, but because they don't pay attention. They're not engaging with the task anymore, they're doing something else. I think that's fair. Yeah? We shouldn't ask people to stay, or fish or mice, to stay focused only on one task to survive in nature. It actually makes sense to multiplex, to multitask, to not obsessively stick only with one. So we should switch. There's attentional switching. That's sort of one of the overarching goals of our lecture today is why do animals stop paying attention? So together with Armin, we have developed a model where we can quantify how fish do in the task, and we find an algorithm that explains what fish will do when they're engaged in the task. And what this model includes is the baseline bout rate, so how fish are swimming before we actually even show them the stimulus. Yeah? There's this baseline bout rate that can go, so 60 bouts per minute would be 1 hertz. Yeah? And sometimes the fish swim a little bit less, the low rate, they're the slow fish, the medium fish, and the fast fish. Yeah? These are just the energized fish, right? The ones over here. And here we have simply a baseline performance score. So this is when we look at the baseline bout rate and ask how fish will do. And what we find is that the slow fish, exactly, slow fish over here perform better than the fast fish over here. So what this tells you, there is already a relationship where you can ask a fish before you even start the trial, if you're slow and meticulous or if you're fast and hectic. You can predict already how that animal will do later in the task itself, in those 30 seconds. So if you're very fast in the baseline, you will do worse than if you take your time. Maybe that makes sense intuitively. If we put this into our model, then it turns out that the model will predict this nicely. So this is a computer model where we model what's going on in the mind of the fish. Here is data from real fish. During the baseline period, performance scores are randomly distributed. Again, here we have the baseline bout rate. Here we have the performance score for all 2,000 trials. Here is maybe the first take-home message. The first, I should spend maybe a minute in. What's plotted on the x-axis is the baseline bout rate. So here are the fast fish, and here are the slow fish. Okay? We see already that the fish who swam quickly during the baseline perform really well. And the fish that swam fast during the baseline, here there's this cone shape, right? This spreads out a bit. But what is much more remarkable about this cloud, this performance cloud, is this real smack gray cloud in the middle. If you look at that, this gray cloud is centered around chance. So all the maybe 500 trials that are in this middle gray cloud, this is the fish not performing poorly. This is the fish performing at chance level. This trial here, so when I first saw this cloud, when Kumar showed me these data, I said, they're not paying attention at all. The fish is actually switching between 2 states. One state where it's engaged, and that's up here. That's the high performance cloud. And then there's another cloud here where they don't pay attention at all. And that, I think, was the seminal, the epiphany. When we looked at that, we say, fish are doing poorly, not because they can't do any better, but they're switching between 2 states. They're either engaged or they're disengaged. And I think that's how we should look at animal behavior. They're either engaged, and then they perform really well, or they're disengaged, and then they perform at chance level. And you only see that if you have 2,000 data points. Initially, we had fewer data points, and we thought that just a few fish did perform less well, the dummy fish and the smart fish. Now we realize there's the same fish can switch between 2 states, one that's engaged and then one that's disengaged. That's basically the insight of the first part of my lecture. I'll show you now data where we try to model this quantitatively. and probably convince you that we can use this model now to describe fish behavior in much more detail, and also to extract now properties of different fish, of mutant fish, of treated fish, and see how this ability to stay engaged or become disengaged, to lose focus, how this can be studied in individual animals or in mutants or in... yes?
So why are you phrasing it this way around? You can also phrase it the other way around. So that it's that the fish is not paying attention.
Yeah.
You mentioned it's a reflex. So it's just the baseline is following the flow pattern.
Yeah.
And that it is, when you call it disengaged, it's paying attention to whatever is out there.
Something else, yeah.
So why did you choose this side and not the other side in phrasing it?
How would you phrase it? What's the other side of phrasing it?
I wouldn't, I'm not working with fish.
No.
We can frame this as well. I think this is a general phenomenon. Well, we can maybe chat about it later.
No, I'm just wondering, I really want to get, what's the question of it?
At the beginning, you mentioned, well, it's a reflex. And then...
No, I don't know what a reflex I have an issue with. Yeah, okay, yeah. It's a reflex, yeah.
And I don't have to pay attention. Then I show this behavior as baseline. Oh, and you're arguing, well, the fish is paying attention.
Sorry, you say attention. Okay, then let's say it's just engaged with the task or not engaged. When I mean attention, I mean it's doing the task, and when it's not paying attention, it's ignoring it. That's what I mean. I don't mean that you can do a task without paying attention. That's what you mean, right? A reflexive task is one I'll do automatically without paying attention. That's not at all what I mean. When I say they don't pay attention, I mean they don't do it. So that's how I use attention and focus, is if you lose attention and you lose focus, I mean operationally, you don't do the task anymore. I'm not talking about whether you need to pay attention while you're doing the task. Yeah? That's what you mean, right? Is that what you mean, that I can do a reflex I can do without paying attention? Is that was your...
Just take the middle cloud,
This one?
Yeah, their decision can happen. The fish can make it.
No, it's not.
Yeah, they swim randomly.
You don't see it, but the way you're arguing is based on the phenotype, based on the behavior.
Yeah.
Well, let's
okay.
do that.
I mean, but that really gets at the heart of it. So my underlying model is that the animal either solves the problem, engages the task, takes the information, and it generates the appropriate motor output. That's my definition of being engaged and doing the task. Or, alternatively, the performance is zero, meaning it's as if the stimulus were not there. Yeah? It's as if the animal is not doing the task at all. That's the behavioral phenotype. Yeah? And then we can argue what's going on, or we can even look what's going on in the brain, and at what level is the motor output now gated off. Yeah? But I think that's a detail, right? The point is animals not doing the task at all.
Just for clarification of the analysis and understanding, because you're talking about two different states.
Yeah.
But the gray cloud is a continuous distribution. So where do you...
Thank you. I'll get to that. I mean, I think this is happening right now. Yeah. So what we do to answer your question exactly, we'll turn this around and we make a fit, right? So we say this is our high performance cloud. Yeah? So this, if we can fit this with a Gaussian, that's our in disengaged state where the fish is behaving as if at chance level. And then we have here a high performance cloud, which we fit with a gamma distribution. Yeah? And then we fit the whole cloud here with a mixed distribution model, which is the sum of a Gaussian and a gamma. This is really important because the ratio between the gamma and the Gaussian here tells us how much the fish is engaged. If it's all Gaussian, then the fish is unfocused, disengaged, and doesn't do the task at all. If it's all gamma, the fish is perfectly engaged and will never lose its attention. Really important because this ratio between engaged to disengaged, we call this focus. That's what's plotted here. So yellow is not focused, blue is focused. And this line can go from 0 to 1. If the blue line is 1, then the fish is perfectly focused. If the blue line, the blue bar is at 0, then the fish is perfectly unfocused. Then it's all Gaussian. The yellow is the unfocused, the blue is the focused, engaged. And the gamma, that's interesting, has a tells you, the gamma part tells you how often the fish is engaged, but the shape of the gamma tells you how well it is doing while engaged, and this gives you an independent variable that we call competence. So a fish, how well a fish is doing depends on two things: on his competence, how well is he doing while engaged, and how often is he engaged. And these are orthogonal, they're independent. So performance can be measured either by focus or by competence, and those two things are orthogonal, they're independent. You can be very focused but incompetent. Like if I try to play the violin, right? I can be really focused, but I'm just incompetent, I don't do it very well. My daughter is spectacularly good at the violin, but her mother tells her sometimes she's just not focused, right? That's why she's playing badly. So those two things are completely decoupled. I think it's really interesting in any animal to ask, when you're doing badly, are you doing badly because you're not focused, or are you doing badly because you're not competent? And I think what we'll do now is look at mutants, look at schizophrenics, look at autistic fish, look at perturbed fish, and see which aspect is changing. Is it the focus that's changing, or is it the competence that's changing? That was really the power of this whole story, is that we can separate now, after a perturbation, which aspect of your performance is changing, is dropping or increasing. Yes?
Is this one fish or is it a group of fish?
This is 64 fish.
And do you consider or pay attention to the temporal aspect of, for instance, they have phases of concentrated focus?
Right, so when you leave them for 1.5 hours, what we'll see that focus, both focus and competence drop down and go down a little bit. So these things can be studied, of course, also over time. You can see how competence or focus changes over time, or how it changes after certain perturbations. This is something you can now extract. Yeah, very good. All right. Are we all on par? Yes?
I have the impression also that during the test, the bout rate is also higher during the cloud when the fish is not doing the task. Is that the case?
Say again? The bout rate?
The bout rate seems to be slightly higher during the test.
Yeah, no, it is. When you give them a task, the bout rate goes up by about 20%. Even if they're disengaged, that's right. So they're not actually less energized, they just don't pay attention to the task. Yeah? That's what we find. That's important. They are not generally phlegmatic and stoic. They just don't care particularly about following substrate motion. They're probably paying attention to something else.
The task is somewhat stimulating them to do something.
To do something, yeah. They are, yeah. All right, so here is focus and competence. Here is how we define it. Yeah, that should be clear. Right? I mean, we can extract, we can quantify. We can quantify focus from 0 to 100%. And we can quantify competence, which in this case, in this matrix, goes from 0 when you're really sucking at it completely, to this one in principle goes to infinity when the gamma function becomes a delta function. Yeah? So competence is not capped really, which might even be realistic, right? If competence is running fast, then it's sort of not capped. There is no. Anyway, that's not really important. So we now model this by proposing two states, one that's engaged, the other one that's disengaged. Engaged, and we just assume very simple. The simplest, it's just a bimodal, a stochastic coin flip that has them flip from an engaged into a disengaged and disengaged into engaged state. And it turns out that model is sufficient to describe all of our data. So we think that the fish is just randomly transitioning, in our particular case, between engaged and disengaged. And now, the first thing that we tested is, are they getting disengaged when they get close to the wall of the dish? That is one of our hypotheses. And indeed, here we have our performance score, and now we are plotting it at distance from the center. Here they are in the center of the dish, and here they randomly swim towards the wall. And we ask, what does this performance cloud look like? And here what you'll see is in the center of the dish, they're performing really well. If they get closer and closer to the wall, then this drops to chance level. Yeah? So this is one way of distracting the animal, is by giving them probably a mirror image. I think when they get close to the wall, these are not sandblasted walls, these are glass walls. They might see the reflection. We don't know. There's a distractor here. That is already interesting. And now we come to the first highlight of our story, is where we leverage the fact that we have two separate fish facilities. one that's in my building, that's the Engert lab facility, yeah, A, where we see the fish are very focused, blue is high, and competence is about 0.6, right, they're competent. Then you have my friend Mark Fishman, has his facility in the Sherman Fairchild building, yeah? And these fish are not that focused, yeah? And they're also not as competent. Yeah? Yeah, this is facility B. Yeah? Now what we can do, we can take fish from the two facilities and raise them in the other facility. So we are asking nature versus nurture. Yeah? What determines focus, what determines competence? Yeah? Is it your parents or is it your upbringing, yeah? The facility? Yeah? Here we have parents raised, parents from A. So what happens if we take our clever focused fish and raise them in the Fairchild facility? And what we'll see, the focus doesn't really change much, but the competence drops. So this suggests already that your upbringing doesn't really change your ability to focus. You get this from your parents, but what drops is the ability to perform in a specific. And now we do the opposite. We take the fish, the parents from B, and raise them in A. And that's what we'll see. Again, competence goes up as expected. Focus goes up a little bit, but not as much as we predicted. Yeah? So it turns out, I mean, that's our current hypothesis, that the ability to focus is largely innate and parental. Certainly true here, whereas the ability to perform while engaged is something that we think is more dependent on the raising conditions. Yes?
Are they otherwise the same? So is it the same strains generally?
No. They're different strains.
They're really different.
They're very different strains. And this is getting now to this. Let me just go through this so you can see this all. So what we did here now is take our fish and now subject them to certain environmental perturbations. These are all different fish strains that we took. In the top one here, we just subjected them to lower coherence strength. We made it harder for the fish. This is just a control, really, right? Is where we'll see. So on the left is control. Here is experiment. Focus doesn't change very much, but the competence drops by 50%, which is absolutely expected. We make it 50% harder. We reduce the coherence. And what we'll see is they don't disengage because it gets harder. What they do, they just don't perform as well. This is completely expected. This is really interesting. If we give them pre-loom, if we, during the baseline, we scare them with looms, what you'll see is the experimental fish pay this. I mean, these show already much more focus to begin with. They probably came from our facility. These are all different parents, different strains, and you see that the ability to focus, as expected, what I told you previously, should vary because that depends on the genetic background, how much they can focus. And you see this again here. The experiment doesn't change the ability to focus. The raising conditions don't change the focus very much. But what we see, the pre-loom makes them pay more attention now, not pay more attention, makes them perform better during the test. This is also interesting if you sleep deprive the fish. Focus doesn't change, but they become better. Their competence goes up after sleep deprivation. We don't know why they're doing better, but it's not focus. It's they pay the same amount of attention, but if subjected, they do better. And here is two. I think this is an autistic mutant. This is a schizophrenic GWAS phenotype. And in both cases, what we see is the genetic mutation does not change the focus. This case, again, the mutant increases its performance a little bit here for the SCN1 lab, and for the other one, we get actually a dramatic increase in performance, and that is that. Yes. Here are the two stars of this paper, and that's what I have so far. We can stop briefly, yes, sir.
What was, I didn't understand the unit of the competence on the plots.
This kappa function.
Yes.
How do we measure that?
Yeah, what's the unit on the y-axis?
It's a little complicated. So the way we fit the performance distribution is with this Gaussian and this gamma function. And this gamma function has a value kappa that tells you how steep, how much squished the distribution is at the one. So it just tells you what is the ratio of ones relative to the smaller than 1 performance indices. It's the shape of the gamma function, which can go from 0, when it basically looks like a Gaussian, this kappa. It can go to infinity when this gamma function looks like a delta function. When the slope, it's the slope of the gamma function is the kappa, right? It's how steep is the performance curve. Yeah? And the kappa can go from 0 to infinity. It's that's
Yeah, before you move on, Florian,
Yeah?
can I just make a comment? I think this is absolutely spot on. We find this in older fish as well, and in ants and in flies, that really it's not a weighting of cues. Actually, it's the weighting occurs via temporal.
Correct.
Like you're either doing one or the other. And then if you model the navigational circuits and you look at the analysis of this, there actually is a phase transition between paying attention to external cues and the network being dominated by internal dynamics. So it's completely consistent with what you're saying. So I think this is a really universal feature of behavior that people haven't spotted before.
Yeah. And I think that the whole idea about having internal attractor states that go to one or the other, it applies here. Yeah. That the animal focuses on one thing, and it's a winner-take-all. And it's a winner-take-all dynamics, right, where they get stuck in one engagement state, and then they can switch into another one where they get stuck. Then,
I have a question about the behavior with respect to the wall, because they're performing better when they're in the middle of the tank. So we'd expect to see, but how much could be explained just by them being closer or farther away to the wall, or what would be? Because an alternative explanation
I think they can't actually turn into the wall because there's a physical barrier. Is that what you mean?
Yeah, so they could be taking the wall into account, and of course that's accounting for some part of this performance decrease.
I mean, there's 2 ways how the wall can play a role. One, it's a physical barrier that prevents them from turning into it. So it's impossible to perform well close to the wall. That's one way, And we can exclude that because they are far away, such they can still, in principle, turn into that direction. The other one would be that the visual image that the wall provides, the reflection, they can see the wall, they can classify it as an object, and that new input switches them out of the substrate-based behavioral state, and that's what we believe is happening. They're distracted by the wall. There's another input that dominates now the behavior and pushes them out of the substrate-based navigation task.
Okay, I see. Switching out of this task instead of weighting the wall and the stimulus.
No, they're not waiting. They're not doing. They actually do one or the other. I think what we believe is that if you're distracted, you're not doing a biased weighting function. What you're doing, you commit. You do either one or the other. I think that's how most mains. I think the main difference is that there's difference between a male and a female brain. I think males can focus on one thing and one thing only, and they will switch between. Apparently, females can multitask. They can keep many tasks in parallel. But I think universally, it's usually true that all animals are much more likely to focus and commit to one task rather than to constantly balance all the different inputs and do a average weighting. I think that's just not how biological brains work.
I was wondering, I think a lot about people in a room clapping. And
clapping?
Yeah, exactly. So if I start to clap after you talk, many people will
start clapping.
Take this as a cue and follow this. So I was wondering if you considered that a feedback where the neighbor reacts to the fish would be actually also able to explain it in terms of the fish is frustrated because nothing happens if he moves. So, for instance, did you do maybe an experiment where you move the dots to the direction or vector of the movement of the fish?
Yeah, no, we did. We did.
Feedback cue.
Right. So all of these fish are, of course, in isolation. If you do the same experiment in many fish, at least 7 days old, right? There's very, very little social interactions at 7 days. I think it's minimal. There's some, but the relationship of social feedback cues is minimal. Also, I will briefly talk about, if I get around, to closed loop versus open loop, whether the fish is frustrated or not. Actually, let me come back to that in a moment. No, we are running desperately out of time, so I will now talk about the heart. And how, and here is maybe how you should think about the next part, the next 8 minutes of my talk, is how we can think of maybe what is switching the state of the fish, whether they're paying attention or not. Maybe it's not what's coming from the outside, but what's coming from the inside of the body. Maybe the fish are listening to their heart, are listening to what the heart is doing, and that's what makes them switch between states. So this is our hypothesis here. The beauty of the larval zebrafish is that we can look at organ states, we can image organs, we can do brain imaging, and we can do behavioral imaging all together. I'll briefly mention the autonomic nervous system, which we share with all vertebrates. We have a sympathetic neurons that agitate the body. We have vagal motor neurons that calm the body. Same in you as in larval zebrafish. They all drive the viscera. And then we have the sensory part. The body also talks back to the brain. In this case, is sensory neurons send information from the body into the brain. This is the nodose ganglion here that we share again with autonomic nervous system. Autonomic nervous system controls the heart. Autonomic nervous system also gets information from the heart back to the brain. And we want to know what is the role of these autonomic neurons in communication between the body and the brain in order to switch between states. That's the talk. Here again is the yours and the fish's autonomic nervous system. Again, we have the motor vagus that drives the heart. We have the sensory vagus that gets information from the heart, sends it to the brain. And we have the sympathetics, which again agitate the heart. We can image heart rate, as shown previously. We can relate it to struggles. This is just showing that we can take the heart rate of a fish, and now we monitor the heart rate of the fish and want to know how does this relate to the rest of the behavior. So first of all, here's the heart rate, around 2.5 hertz. This is actually, no, this is 0.5 hertz. This is the heart rate of the animal. And if we agitate the animal, either with a salt pulse or with a dark flash, these are all aversive stimuli, then we can see that the heart rate transiently goes up. Sometimes it also goes up spontaneously. Then we don't know what's his problem, right? Sometimes the heart rate just goes up and we don't know why, he's just unhappy or whatever, right? But we can also do evoked heart rate elevations, and that makes sense. This is just an external aversive stimulus. 15 minutes is the time scale here, all right? This is, I think, a readout for internal state. If you want to know how happy that fish is, how scared or how agitated, just look at the heart rate, and it tells you already something about its internal state. It's an internal state decoder. And actually, this is not for the people who have issues here. This is actually a truly spectacular plot that Christian Herrera found. This is spontaneous heart rate fluctuations, and these very faint blue and red dots tells you whether the fish is engaged or disengaged, whether it's doing the OMR or whether it's not doing the OMR. Each of these vertical stripes is a trial. And what you see here already is that when the heart rate is up, the trials are at chance levels, unresponsive OMR trials. It's a little bit more in here on white on black. So this is one of the take-home messages. Is unresponsive trials correlate to elevated heart rate. So it seems when the fish is not paying attention in our previous story, maybe it's just his heart rate went up for whatever reason. And the rest of the talk, I'm going to talk about what...
The heart rate could go up because the fish is not paying attention.
Well, I'll get to that. So who is what's the chicken and what's the egg here, right? So let's get into that. First of all, what we'll see here is a linear relationship between heart rate and responsiveness. So this is really very reliable. This is true. I mean, there's no argument. And the latency, how long they take to respond, again, depends on heart rate. So this is really performance. The performance index really relates to heart rate. And now I'll briefly introduce the two. Maybe Ian alluded to that already. There's two different ways how we can think about this. It's called the Cannon-Bard versus James Lange theory. It's actually kind of cool, right? So Cannon-Bard is the classic theory. If something scares me, this green arrow gets into the thalamus and then generates some problem. And that leads to the heart rate going up through the sympathetics, and it changes my internal state. This is sort of common origins problem of internal state. James Lange, William James, and Paul Lange, they came up with an alternative theory that the aversive stimulus enters the thalamus drives the heart rate up, And the fish is getting scared, not because of the threatening stimulus, but it's just listening to the heart, and that's why it's scared. This is the idea that if we are unhappy or scared, we're not scared because of the bear, we are scared because our heart rate goes up. And this is illustrated much better here. Yeah? So we have a stimulus, I see a threatening bear, and Ken and Bart would say that threatening stimulus drives my heart rate up and drives my internal state up. James Lange, I think is interesting. That's an interesting alternative. Here, I'm scared because I'm running. The previous one, I'm running because I'm scared. Here, I'm running because I'm scared of the bear. In the second case, I'm scared because I'm running. I'm scared because my heart rate is up. I'm scared because my body responded, and that gives me my emotional state. This is what we're going to test in the last 3 minutes. Is this true? Can we induce a change in the state of the animal by just bringing the heart rate up without any other stimulation? If we just bring the heart rate up of the animal, of you or of the fish, will this be sufficient, necessary and sufficient to just change the emotional state of the animal? That's the test we can do now in the larval zebrafish. And the way we do that is we can optopace the heart. This is my one use of channel rhodopsin, is where we can put channel rhodopsin into the cardiomyocytes. And we use a laser pulse now to change the heart rate frequency by just optopacing the heart. And what we can do, we can clamp the heart rate now. We can make the heart rate high or low. We can determine just by a light pulse. So nothing else is changing for the animal. We can just now artificially bring the heart rate up or down. Yeah? This is the individual heartbeats, and we can just clamp them. And here is the behavior now. Yeah? And our prediction would be that this is just a shitty ass plot from Christian. We'll look at the behavior later. So the prediction would be that if we are pacing the heart high, the OMR should go down. And there's lots of controls, and it's all significant. But here is what we should focus on. Yeah? In a fish with no channel rhodopsin inside, pacing doesn't do anything to the OMR performance. If we put channel rhodopsin into the cardiomyocytes, then whenever we go to 2 Hertz, performance drops. When you go to 2.5 Hertz, it drops even more. So we can induce a behavioral internal state of the animal. We can get it to disengage just by bringing the heart rate up. And finally, we want to now look at the implementation. How is heart rate communicated to the rest of the brain? That's the question. Yeah? How does this information from the heart rate with optopacing here? It's probably the nodose ganglion, because that is the nucleus that listens to the heart and sends information onward. How is this relayed now to the rest of the zebrafish's brain? Yeah, the nodose ganglion is right here. We can label it. We can do calcium imaging. And we can find that the nodose ganglion indeed tracks heart rate. Yeah? That's nice. That's a given. I mean, we thought that would be the case, but indeed we can find that the nodose ganglion indeed has the neural information to relay heart rate. And now we want to know, where does it go? Where does it send its axons? For that we use connectomics. And we can do the nodose, complete nodose connectome. We can look up in the brain where it's synapsing. And one of the targets we found are astroglia. And I'll end the last 5 minutes, I'll spend on the potential role of astroglia to take information either from the heart or also from the outside world, frustration signals, and how astroglia can probably store this information for many minutes, can integrate this information, and can even use this information to influence the behavioral state of the animal. This is going to be the final. So what is the functional role of astroglia? Here we'll have a 3 to 4 to 5 minute intermezzo, the final intermezzo. I'm going to talk about the futility tracking system. This gets a little bit to the question that came from the back previously. What if the fish is frustrated? What if it's in an open loop environment where its actions don't lead to the desired consequences? This is what Misha Arens, who developed all of that, this is the assay. So we have a head-fixed fish now, which is responding to a moving stimulus. And what you can see, this is a closed loop setting, where whenever the fish swims, it acts, the stimulus responds appropriately. This is a happy fish that is not frustrated. Yeah. Now we can frustrate the fish, and whenever it swims, we keep the bars moving. This is a frustration. This is when your actions do not get the appropriate feedback. And what happens then, if we go from closed loop, where the fish swims along normally, if we go to open loop, fish starts to get unhappy and struggle. And after a while, the fish will struggle, and then it's going to give up. That's a very reproducible, it becomes passive. And this transition from closed loop to open loop to becoming passive is something we'll explore. So first there has to be an error signal. Look at these neurons here. What Misha and my lab also found is exactly how this information is represented and relayed through the fish brain, and probably also the mouse brain. First of all, you need to find a mismatch signal, where the swim command is suddenly not matched anymore with the sensory feedback. There's a mismatch, yeah? I'm swimming forward, I'm predicting there should be flow backwards. If it doesn't come, the predicted flow backwards, I get a mismatch signal. Yeah? That's in these noradrenergic neurons in the medulla oblongata. We can measure that. We can find them. These pink neurons acutely report any mismatch. Then we have an error accumulation. If mismatch keeps coming, it gets accumulated. There's a green accumulation signal that lasts for many, many minutes. Yeah? Over minutes, it accumulates. And then ultimately, if that reaches a threshold, that green signal, we get suppression. And where does this accumulation, this long time scale accumulation happen? Well, that happens in glia cells. Yeah? I call this system the futility tracking system. Yeah? Or short, what the fish is thinking. Yeah? When something happens again and again and nothing works, you say, fuck this shit. Right? Yeah? FTS is the futility tracking system. Yeah? Futility tracking system, that's the appropriate name for it, but the mnemonic is, you know what the mnemonic is, why this makes sense. I'm just showing this now. This is one of the, can you shut the lights off? This is one of Misha's movies where the fish is in closed loop. Everything's going fine. On top, we have all the neurons that are active, yeah, all the brain areas, while the fish is swimming along in open loop. No frustration, standard going. In the bottom, you had the astrocytes, which were silent. And now we go to closed loop. And wow, this is frustration signals in the glia. The glia are now lighting up, and this is pocket, right? This is frustration, yeah? And when the glial signal reaches a certain maximum, see the time constants here, while these are high, fish is not doing anything, yeah? This is played back 3 times higher, and whenever there is a struggle and a frustration, glia signals goes up. And those glia is, I think, they govern these long time constants. They can release ATP, signal to GABAergic neurons, which shut down the motor command. And okay, now let's. This is one of the final slides here. Yeah. So we have first failure detection. Yeah. Acute failure, mismatch detection, frustration signals. Then we have over time. These glia cells slowly, incrementally ramp up, get more and more and more and more. And ultimately, they reach a threshold and suppress the swimming. And finally, if that's true, if that's our model, this would mean that astroglia, if they are the ones that relay heart information, then we should image an astroglia and see whether they actually track heart rate. That was the critical experiment. If that's true, then let's image the astroglia, and if you're right, then they should actually track heart rate. And look at that. Do they ever track heart rate? This is the heart rate. This is what the glial signal that we find. And it's almost uncanny how well this works. I mean, this is, to me, really nailed it, that the astroglia are the ones that sort of represent the internal state of the animal, and that can, where the rest of the brain is listening and deciding which problem to engage in. And now finally, we can also optopace and see whether astroglia, so with channel rhodopsin, and indeed this works. And I think this is where I'll stop. One last thing that I want to end up with here is that this idea that internal states, in this case it's being stressed, being frustrated, that's not being stored in recurrent neural activity, which is very, very hard to do. Neurons have time constants of 10 milliseconds. To store this information in neural recurrent activity is difficult. I think what evolution did, it found a cheap hack. It found a trick where it used the low-dimensional space of glia, which are really bad at fast computations, and they're really bad at high-dimensional representation, but they're really good at long-time storage, right? Because that's the intrinsic dynamic. And what evolution found is that they use these shitty-ass glia cells that can't really do much computationally, but they're good for intermediate storage for minutes, up to minutes. And what happened now is that that particular aspect of the computational problem is outsourced to glia cells, which can now store this information for a few minutes. And I think that is interesting. It's a hack, but it works really well. And if you know that, then you can also see where you have to look to if you want to see how these long-term changes, these low-dimensional changes in internal state, how they are being governed, how they're being driven, and how they're being read out by the rest of the nervous system. And with that, I'll stop and take questions.
So thank you so much for this really engaging talk across so many levels, which all come together beautifully. Are there additional questions?
Only quick question about the chicken and the egg there. So I think, I'm not sure, but I think what Ian meant was whether it's the other way around, is whether the fish is frustrated and then the heart rate goes up.
In the second case, right? No, I think that's true. No. I think that's Ian's right. In this particular case, we believe that the mismatch signal between the efference copy and the returning feedback, that is actually computed in the raphe, in the medium raphe, and that medium raphe signal, that frustration signal, goes to the sympathetic neuron and drives the heart rate up, and also goes to the glia cells, right? So in this case, it's coming from the outside. The frustration signal can come from the outside and drive the both signals up. This is more like cannon bar. But I think what we realize here, that if the heart rate goes up for another reason, that's purely internal, right? The oxygen deprivation or something that is something, or blockage or cardiovascular, whatever, that will also lead to the same phenotype, the anxiety that now doesn't come from the outside, but comes from the inside. What the important part here is that heart rate alone is sufficient. That's exactly. But who's the cause for the heart rate? That's another topic of the talk that I don't want to get into, but that's something one can also study. How is the sympathetic system involved in driving up the heart rate, in controlling the heart rate? And are these external cues or internal cues? And it can be anything, really.
Thank you very much for giving us lots of food for thought. I have a question on the glia cells and the connection. So are you suggesting there is a direct connection or neural connection to the glia cells, or could it also be that the glia cells essentially monitor neural state, and one could also see when the fish's signal was mismatched that they generally got much more active, that also the neurons in the brain got somewhat more active, and that this activity might trigger the glia cells to also start metabolizing or doing something, and kind of being a consequence of neural activity rather than the specific mismatch. So
so the mismatch is neural activity. So it's neurons in the middle. Someone needs to compute the mismatch. And that's a neuron, right? Absolutely. So the glia are listening to neural input. Or are you suggesting that they have an independent way of getting the mismatch that's not neural, as an alternative hypothesis?
I'm more asking, is this a specific function, or do the glia cells, as we know, have more of a, let's say, maintenance and supportive function, and whenever the neuronal activity is really high, whether it's a mismatch or whatever else it is, the glia get active and start their kind of regulatory function.
That is the classic. That's the classic party line. Glias are for maintenance, and if neural activity goes up, glia also ramp up to keep up with the maintenance. What Misha found is that that's just not true. That glia can directly listen to noradrenergic neurons. There's a pathway from noradrenergic neurons that we have found, and we know the receptor, and we know exactly that glia, in addition to looking at homeostatic maintenance stuff, they have another role, a much more specialized role, where they have dedicated receptors and they listen only to noradrenergic neurons, which are the stress generators. So glia cells can listen to these neurons, And what we know also is that when those noradrenergic neurons who broadcast elsewhere as well, when they are active, then glia integrate. They're not just linearly, they're integrating this noradrenergic information. Really, they're perfect integrators. And when they reach a threshold, this is a paper that just came out this week, I think, we know now what neurotransmitter glia use to talk to the inhibitory neurons. Turns out to be ATP or adenosine. And GABAergic neurons have an adenosine receptor that's blocked by caffeine, which is interesting. That's why if you drink coffee, you're not blocked, you're excited. Also, there's a ketamine story about this too. But glia talk with adenosine to inhibitory neurons. So this whole crosstalk between glia and neurons now is figured out. So we know really what the language is with which they speak to each other. The only difference with glia is they speak 100 times slower. And they can integrate and remember something that's really hard for neurons. These time scales of minutes to Ahmed and I have discussed about this. Right? That's really one of the hardest problems in neuroscience is time scales of minutes. How do you store this information actively in a neural network? That's really, really hard. And the glias can do it effortlessly. It's almost cheating.
I think this is really amazing. But the two mechanisms you propose, they don't seem to be mutually exclusive.
Correct. You mean the Cannon-Barth and the James Lange?
One would imagine that actually both mechanisms simultaneously would perhaps be more effective than each one in isolation.
So it's funny that you say that, because this was a huge fight in the psychology community. And as with all of these huge fights, here, James Lange, Cannon-Barth. Really, I mean, vicious. I think they're still fighting. And in all of these big battles, as it turns out, they're probably both right? They're both, it depends really. So nobody would seriously argue that if you're running away from a bear, that you have no idea why you're frightened. It's just your heart, right? And that it doesn't have anything to do with this. So I think that clearly, the thalamus doesn't only talk to the heart. The breast of the brain doesn't only listen to the. There is, of course, also an arrow that goes from the thalamus to the amygdala, the periaqueductal gland, the cortex. So that they work in parallel. I think the only reason why I've belabored this is that this is usually neglected.
I completely agree, and I think that's really important. And then in your experiment, which I thought was really beautiful, did you also do a sort of control where you lowered the heart rate?
We did, but we can't. The optopacing doesn't work for lowering the heart rate because the pacemaker takes over then, right? So if you don't stimulate, what we did do, we used drugs. We used beta blockers and propranolol, which is also used for nervous grad students when they prepare for an exam. And you can take a beta blocker, which is a noradrenergic receptor blocker, which also lowers the heart rate. And if you do that, then the fish never disengage. Cool. So if you give them beta blockers, then like students, they perform much better in the exam. They will then not get nervous and neglect. So we used pharmacology to lower the heart rate, and there we find that the first, the glia cells stay silent, and more importantly, the fish never disengages with the.
That's interesting for another reason, in that the drugs that students often take as study drugs actually don't have any benefit in performance. It has perceived benefit.
That's the placebo.
It doesn't have any quantitative benefit. The other thing is I mean, this is now just taking it further, so not for this age zebrafish, but I find this really compelling to start thinking about physiological synchronization between individuals driven by social interactions, like heart rate variability synchronization, for example, and whether the zebrafish might be also a good model to sort of think about, humans, for example, exhibit this phenomenon, and this could be something very primal.
That's right. I mean, synchronizing internal states, right? That if animals need to, even for collective behavior, it probably helps if all the collective is engaged in the same problem, and if not, one half solves one and the other. So for what to focus on, if you decide to pay attention and to focus, it helps if that ability to focus is collectivized.
Thank you. You already given half of the answer to my question because I wanted to ask about the beta receptors. What kind of receptors are on these glial cells? Because typically beta blockers, I explained more that they act on the heart.
No, sorry, the glia don't have...
And the question is, would the same beta blockers also act on the astrocytes? And then you have a dual action of the beta blocker.
The glia cells, I know, they have the same receptor. It's an adenosine. This is not the glia adrenergic receptor. The glia cells have an adrenergic receptor, which I don't know whether it's the same as the one that's on the heart. This is known. It's just I don't know it. Yeah. So one can easily.
But in your pharmacological experiment, when you use beta blockers
to block the heart.
Right.
I don't know whether that
be that it also.
No, I think that's known. I think it doesn't, and I think that's known because the receptor on the glia cells is not the one that is susceptible to propranolol. I think that's known. Again, I don't know which one it is and which subclass alpha, but that's something one can. I think that
Perfect. That was the second half of my question.
I just want to verify if this is, as I think, maybe related to attention deficit hyperactivity syndrome. So in the end, if your model would be right and translatable to humans, would this mean that maybe the children have a high heart rate in certain phase and that's why they cannot pay attention?
Genau, that's the idea. Yeah. So if heart rate is related to attentional switching, then just bringing the heart rate down. There is some anecdotal evidence for that. It's about meditation. So if you want to calm down and become more focused, you meditate, and that's what yogis can do. They can actually actively, by meditation, bring their heart rate down, and that has beneficial effects in terms of focus and attention. I think that's probably maybe a much more preferred method than taking beta blockers, right?
Okay, thanks again for this wonderful talk, this discussion, and thanks again for coming, and have a good day.